The year was 2015. A group of tiny flatworms was launched into space onto a NASA rocket ship. The goal was to study the effects of the extreme environment of space on their natural abilities. But these weren't just any worms. These were planarians, nature's master regenerators. Planaria are a type of flatworm that are famously known to have the ability to regenerate organs at both their ends when cut. They can even regrow their entire bodies out of tiny fragments. When cut in half, they become two. Cut into 100 pieces, you get 100 new worms. Their regenerative ability is unparalleled, yet their genome is noisy, chaotic, and filled with mutations. They should struggle to even survive, but somehow they defy genetic limitations and rebuild themselves flawlessly. And on this space trip, they would be kept in special biocontainment units and monitored for spontaneous regeneration. Under space conditions of microgravity, radiation, and altered geomagnetic fields. But before the launch, scientists removed their heads and tails, expecting them to regenerate as they normally would on Earth. After 5 weeks aboard the International Space Station, the worms returned home. But something strange had happened. Among the 15 amputated fragments, one had defied all expectations. At the cut end of its abdomen, where it was supposed to grow a new tail, it had instead grown another head. This anomaly had sparked curiosity among many scientists, but to one man it felt familiar. Leaning forward in his office chair, Dr. Levin peered excitedly into the computer and squinted at the report from the space agency. Levin paused as the data loaded. It reminded him of something. Years earlier, he had done something eerily similar. He had been studying frog embryos, textbook biological models, nothing too unusual, until he began looking at something that most scientists ignore: electricity. By manipulating electrical patterns inside embryos, he had guided cells to grow into body parts in places where that organ had never belonged before. He hadn't changed the genes, just the electrical signaling. So when he saw a worm grow an extra head, Levin didn't see a fluke, he saw a familiar pattern. Maybe it wasn't the radiation, or even chance. Maybe it was a glimpse of a deeper code, one that he had been chasing for years. A decade ago, Levin had begun exploring an idea that most biologists weren't even looking for. Long before anyone launched worms into orbit, Levin had stumbled onto something strange: that the body might be guided by a hidden code, not made of molecules, but of voltage. Not shocking bolts of firing neurons, but gentle gradients of charge that flowed across developing tissues. He had discovered that cells in a developing embryo carry tiny electrical charges. As the embryo developed, certain areas lit up with unique electric signatures. And those charges weren't random, they formed certain specific patterns across the body. These were deterministic maps, like blueprints, written in electric current. That alone was fascinating, but then came the twist. They weren't all charged the same. Different regions had different electric signatures. Where an eye would eventually grow had a specific voltage, but the tail region had a different one. Each future organ from head to tail had its own voltage pattern. And here's what stunned him. These electrical maps appeared before the genes for those organs were even switched on. In other words, electricity was talking before DNA even entered the conversation. Levin began to wonder, Was this mere background noise? The lack of any chaos, but rather a well-ordered structure to his signals, made it easy to dismiss that idea. That left two options on his plate. That the subcellular sparks were either a biochemical result of organ development or, very weirdly, a control system guiding it. So he began to tinker, and what happened next wasn't subtle. The results were astounding. Skin cells transformed into neurons, tissue twisted into wrong shapes, cells began building things they were never coded to build, and organs began forming in places they had never belonged. No genes were edited, and no instructions rewritten. All he had done was change the voltage, and the body responded like a machine that had been rerouted, like a GPS that was being given new coordinates. The Levin lab found itself testing a novel hypothesis. Could it be that cells don't just follow their DNA, but actually respond to physical signals as a biological GPS? Levin started seeing the body as an organic device running on electrical software. A software that did not just influence development, but actually directed it. So when he saw a worm grow a second head in space, he didn't see a freak accident. He saw a pattern. It wasn't a mutation, but a rerouted signal resulting from a voltage glitch. There was no radiation at play here, nor microgravity, only some misplaced software instruction. To him, the worm wasn't broken, it had simply followed a different signal.